Hello, good morning, everyone. It's my pleasure to give this lecture. Um, let's start with chapter 1, Introduction to the Microbe and their Building Blocks. In this lecture, we are going to study the various type of microorganisms and those can colonize humans, describe the role and the impact of microbes on the earth, and the theory of evolution, explain the ways that human manipulation organisms for their own uses, and summarize the human diseases caused by the microbes, differentiate the bacteria, archaea, and the eukaryotic microorganism. Okay, so what is microbiology? Um, probably you guys know biology, right? So the biology studies huge living organisms such as elephant, wolf, and human. Okay, so the microbiology is subject to study the tiny living organism. So these living organisms are very small and tiny, and so they are too small to see by the naked eye. Okay, so microorganisms include bacteria, archaea, protozoa, fungi, helminths, viruses, and algae. So the microbes are very easy and difficult to study. Uh, so because they are easy to study, because the microbe can reproduce rapidly. So it takes about minutes or hours for the microbe to generate to produce the next generation. Okay, so it can grow in a large population in a lab. So the scientists in the lab can use the flask to grow tons of microbes. So the microbes are also difficult to study because they are too small to see. So the scientists have to use a specific device to observe the microbe, such as the microscope. Okay. Um, so such as the microscope. Okay. Like this microscope. So we will talk about this later on. Now let's do this question. Which of the following is not considered a microbe? So I give you guys 5 seconds. Double check your slides and give me the correct answer. Okay? 1, 2, 3, 4, 5. Okay, so the correct answer is C, mushroom. Okay, because the mushroom is it's not a microbiology. Okay, it's not a microbe. It's so it's a relatively huge object you can see. So as you can see this object, so it is not a microbe. Okay. So let's talk about the relation between the microbes and our planet, the Earth. Okay. So the microbe uh, play a very critical role to shape our Earth. Okay, for billions of years. So the scientists found that the single-cell organism appeared on this planet about 3.8 billion years ago. So it is the time start of the living organism. Okay. So until now, so we have 3 type of single cell. Okay, single cell system. So they are eukaryotes. So eukaryotes are the true cells. So when we talk about the true cells, that means the cells contains the true nucleus. Okay. So the bacteria is the single cell. Bacteria is the prekaryocytes because the bacteria doesn't have the nucleus. Archaea is also a group of bacteria. However, this group of bacteria exists a long time ago. For example, for billion years ago. So those bacteria lived billion years ago. They are called archaea. So now we have this diagram that shows these three groups. Okay, so for now we can also find the archaea. However, those archaea lives in extreme condition environments such as deep sea or the volcanoes. Okay. So bacteria and archaea are the single cells. So, however, eukaryocytes. Eukaryocytes are also single cells. However, tens of eukaryocytes can link and conjugate together to form tissues, organs, and to form huge living organisms such as elephant and human. Okay. So this is the evolutionary timeline. Okay, so we can see here 4 billion years ago. So it's actually 3.8 billion years ago. So it's the time to emerge the bacteria. Okay, so before this time point, actually, it's a period that this a period that the Earth didn't have any living organism. Okay. So actually, if we look at this timeline, we can see. So most of the period are so the bacteria actually exist in most of the period. Okay, from the 4 billion years to 2 billion years. Okay, 2.5 billion years, and then after 2.5 billion years, so the eukaryote emerged. Okay, and then so we have the insects and dinosaur and mammals. Okay, actually the human only take a place of a very tiny period in this timeline. The history for human is only 40,000 years. Okay, so we can see here. So the microbes are ubiquitous because the microbes are everywhere. Okay, so they are in the water, in the ocean, they are in the mountains, they are in the air. Okay, so when we take a breath, so we can breathe in thousands of microorganisms. Okay, even in the north, even in the polar ice, so there are tons of microbes. Okay, so so the scientists have generated the theory of evolution. So actually, that's Britain scientist Darwin who postulated this theory. Okay, actually, so the theory of evolution has been proved and documented every days and every years. Okay, so we have more and more evidence to support this theory. Okay, so now this theory is widely proved and accepted by the scientific community. Okay. So the theory, so the term of theory in science, that means so actually the theories come from hypothesis. Okay, if the scientists find questions, so they will so they will so they will give their hypothesis for the to answer the questions. Okay, and then the scientists will do the experiments or to do the study to find the evidence to support their hypothesis. If their hypothesis is proved to be true, then so this is can be the theory. Okay, so that means the theory has has been tested for years. Okay, and there are many evidence to support this hypothesis. So actually, so this theory answers the so the theory gives answer for the bear study and bear established nature for phenomenon. Now let's take a look at this question. Okay, the theory of evolution is not widely accepted among scientists because it still remains unproven. So I give you 5 seconds. So go back to collect the slides and find the right answer. 1, 2, 3, 4, 5. Yes, the correct answer is B, false. Because the theory of evolution has been proven by many evidence. So this theory is widely accepted and by the scientist community. Okay. So the microorganism actually is very specific because they have their own strategy to produce the materials and energy. Okay, so so their strategy is photosynthesis. So many microorganisms can use the sunlight to produce the materials and supply energy for their life. Okay. So actually, the photosynthesis is to utilize the sunlight to convert the carbon dioxide and water to the organic material, such as sugars, proteins, and lipids. In the meantime, so photosynthesis can also release the oxygen for us to utilize. There are 2 types of photosynthesis. One is the anoxygenic photosynthesis. This type of photosynthesis doesn't release the oxygen. Okay, so they can use the sunlight. They can also make the organic materials. So the second is the oxygenetic photosynthesis. So this type of photosynthesis is the major type of the photosynthesis. So as we know, so the trees and the grass, so they have this type of photosynthesis. Okay, actually, so the microorganism produce 70% so I respond for the 70% of the Earth's photosynthesis. Okay, that means the microorganism provide 70% of the oxygen in this Earth. Okay, so the trees and grasses only provide 30%. Okay, so we can see the microorganisms are very important for our human and animals' life. Okay, so if without microorganism, So we don't have enough oxygen to breathe, correct? So let's talk about how, so why are the microbes important for our planet? Okay, because the microorganism can produce some of microorganism, so they don't have the photosynthesis, so they just digest the organic materials to produce and then utilize the energy from the organic materials to release carbon dioxide and so this actually so the nitrogen oxide, so this gas actually so they can form our Earth so our Earth's atmosphere. Okay. Actually, so the microorganisms are the most abundant living organism in the ocean. Okay, yeah, so in the land, so in the lands. Okay, so bacteria and fungi live in close association with the plants to assist to provide the nutrients and waters for the plant. Okay, even for the animals and humans, because in our colon, so there are tons of bacteria such as E. coli. Okay, so those bacteria actually provide nutrients such as vitamin B and vitamin K for our human. Okay, uh, so especially for the vitamin K, because our human cannot synthesize vitamin K by our body. Okay, so we can see the bacteria is also important for humans' life. Now let's take a look at this question. Microbes are found everywhere on the planet, except in the deeper layer of the Earth's crust, which are largely considered to be sterile. So I gave you 5 seconds. Take a look at the previous slides and pick the correct answer. 1, 2, 3, 4, 5. Yes, correct. So the correct answer is B, false. Okay. Yeah, because the microbes are very even in the deep layer of the Earth's crust, oceans, and skies. So they are everywhere. Now let's talk about the historic use of microbe by humans. Okay. So our human has known the microbes functions for a long time. Okay. So we use the bacteria to make the bread. Okay. So we use the yeast to make the puff of the bread. Okay. We also use yeast to produce alcohol. Okay, because the yeast have the fermentation function. Okay, to digest the sugars and convert sugars to alcohol. We also use the bacteria to make cheese. Okay. Sometimes, so the scientists also use the bacteria to mine the metal. Okay, for example, some bacteria can eat the gold. Okay, because it's difficult to mine the gold, right? So the people can use the bacteria to let the bacteria grow on the mine so that it contains the gold, and then so the bacteria can eat the gold. Then the scientists can collect those bacteria. Then they have the pure gold. Okay, that's very cool, right? Also, some scientists also use the bacteria to clean the pollution or contamination. Okay, for example, for the petroleum industry, the petroleum industry can cause the petroleum pollution, right? So it's very cost-consuming and time-consuming to clean those petroleum pollution. Some scientists isolate and generate some cruel microorganisms. So those microorganisms can grow in the petroleum, digest the petroleum, and use energy from the petroleum. So they can clean the petroleum pollution. Okay. Now let's talk about the biotechnology. So this is a very cool area. Okay. So since the scientists found the DNA structure and the protein it's a protein. Okay, so the scientists use the technology to 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 adjust the DNA of the proteins. So they generate a new technology. So this is biotechnology. So that means biotechnology contains the technology to manipulation the genome. Okay, so we call the genetic engineering. So they can also, so the genetic engineering, that means so they can delete or insert certain fragment of DNA to the genome. Okay, so that means so they can modify the genome. So this is called a genetic modify organism. So the scientists use this technology to generate a new living organism, such as the a transgenic corn, transgenic potato. Okay, so you guys can find these products in the grocery, right? In Walmart. Once you find these products, actually these products are labeled with GMO. Okay, GMO. That means those are the products modified by the genetic engineering. Okay. The second, so the scientists can transfer genetic material of one species to another species. Okay, so this is recombinated DNA technology. Okay, so by using this technology, so the scientists can also generate new living organism. Okay, so third is bioremediation. Okay, so we can use the bacteria of the microorganism to clean some toxic toxic pollutant. Okay, for example, I have mentioned that for the petroleum industry. Okay, so the scientists have already used bacteria to digest the petroleum to clean the pollution. Okay. However, so the microorganism can also cause trouble to human. Okay, so they can harm a human. They can cause disease to human. Okay. So those microorganisms that can cause disease to a human, we call those as pathogen. Okay, that means those microorganisms can cause diseases. Until now, so the scientists have found over 2,000 different microbes can cause diseases. Okay, so annually, 10 billion new infections occurred in this world. Okay, So do you know how many people living in this world? Yes, actually is 7 billion. Okay, 7 billion. So 10 billion divided by 7 billion. That means every people normally every people will get 1 or 2 infections every year. Okay, so that's a lot. Okay. So infectious diseases actually are the common cause of the death worldwide. Okay, so they cause many people dies every year. So this diagram tells us how many people dies caused by the infections. Okay, so we can see here. So the number one cause for death is the heart disease, and then second is cancer or stroke. Okay, so for United States, so second is cancer. Okay, so we can see. So this is respiratory infections. Okay, so worldwide, so this is number 4 cause. Okay, for United States, it's pneumonia and influenza. So that's eight. So that's eighth cause of the death in United States. So we can see. So actually, so this is number of deaths. We can see many people died on the pneumonia infections. So the microbe can cause very severe diseases, such as AIDS, HIV, okay, hepatitis C, okay, can cause hepatitis, Zika virus, okay, and tuberculosis. In United States, tuberculosis is very rare, okay, because the United States has the best public health condition and healthcare facilities, so the tuberculosis is very rare. With the development of the technology, the scientists also found that some diseases are also induced by the bacteria. Okay, for example, the gastric ulcers. Okay, before the gastric ulcers was considered as non-infectious diseases. Okay, however, the scientists isolated one type of specific bacteria from gastric ulcers. It is called Helicobacter pylori. Okay, so this type of bacteria can live in the stomach to cause the ulcers. Okay, so now, so the gastric ulcers is considered as one of the is considered as infectious diseases. The other diseases such as multiple sclerosis and coronary artery disease and obesity and obesity are also considered also found the tight relationship with the chronic infections induced by the microbe. Okay. Um, so the scientists found the microorganism can cause the disease, so so the scientists also generate and discover many drugs to treat this microorganism. However, the microorganism can generate their capacity to resist to the drug. Okay, so that means the scientists have to study and generate new generation of the medications to treat the microorganism. Now let's take a look at this question. Diseases such as heart disease, gastric ulcers, diabetes, and multiple sclerosis have microbial cause. So I gave you guys 5 minutes. Okay, you can go back and take a look at the slides and find the correct answer. 1, 2, 3, 4, 5. Yes, the correct answer is A. So, oops. So that's true. Okay. Yeah. So now we have found that bacteria, microorganisms can cause those diseases, such as heart disease, gastric ulcers, diabetes, and multiple sclerosis. Now let's talk about the cellular organization. Okay, so there are 3 types of cellular system. The first is eukaryotes. So first is the eukaryotes. Okay, so the eukaryotes means these cells have the true nucleus. Okay, so they also have the complicated organelles. Okay, the organelles actually means the cellular organs. Okay, those are the membrane bonded structures. Okay, so they have specific functions such as provide energy and synthesize the proteins, synthesize all kinds of hormones. Okay, so the other 2 groups are the bacteria and the archaea. Okay, so those cells are the prokaryotic cells. Okay, because they don't have the true nucleus. So they also don't have too many organelles. Okay, so also so the size of these cells are very small. That's why they are called microorganisms. So we also have one type of the microorganisms. So these are the viruses. Okay, actually viruses are not the cells. Okay, they are the they are the living particles. Okay, living particles. So they are not a living cellular organism. The viruses contains protein. So the viruses contains the protein shell. Okay, in the protein shell, so there are either DNA or RNAs. So those contains hereditary information. Okay, so the viruses also are ubiquitous, so they are very rare. Okay, so they are very, very tiny compared to the microorganism. Now let's take a look at these questions. This question. Place the organisms below in order from largest to smallest. So I gave you 5 seconds. Try. You guys can take a look at the slides and find the correct answer. Okay, the correct order. 1, 2, 3, 4, 5. Yes. So we can see the biggest one is D. Okay, biggest one is D, and then is protozoan. Protozoan is second. So the protozoan is smaller than the humans. So the third is the bacterium. Bacterium is smaller than the protozoan. So the smallest is the virus. Okay, smallest is the virus. Virus is very, very tiny. Now let's talk about the history of microorganisms. Okay, how the people discovered the microorganism. Actually, the scientists discover microorganism from 16th centuries. Okay, so the scientists invented the microscope to visualize the microorganism, and then so we have the subject of microbiology. So the so for the history of biogenesis, actually, so the scientists believe that scientists believe that the invisible vital forces makes to create the living organism. Okay, so so for example, so the molecules such as carbon and so the molecule of carbon dioxide and the water molecule, so these molecules conjugated together and then form the organic material. So the organic materials also meet together to form the complex complex macro molecule, okay, such as DNA, protein, or lipid. And then those macro molecules form together, link together to form the cells, and then so forth. Okay, so the tissues make the organs, organ make the living organism. Okay. Um, so so the scientists also believe that only the living things can generate the living things. Okay, only the living organism can produce the living organism. So, for example, if we have, for example, we can see flies generated from the from the earth. Okay, so actually, so this fly eggs produce the fly. Okay, it's not the earth produce the fly, right? So this is the biogenesis. Now let me introduce Louis Pasteur. Louis Pasteur is a very famous scientist. Okay, so he was a very smart guy. Okay, because he had many inventions. Okay, he firstly studied the role of microorganisms in the fermentation of beer and wine. Okay, so when he studied the fermentation, he generated the aseptic technology. Okay, we call this pasteurization. How he generated this technology? Because he did experiment. Okay, he make a flask. He made two type of flask. He filled the flask with beef broth. Okay, he for example he made these two type of flask with the swan neck. One for one flask, so one flask doesn't have the swan neck. So the pastor heat and boiled the broth, okay, to kill the bacteria in the broth. Once he broken the swan neck, so the bacteria can get into the flask to contaminate the broth and to make the and the growth in the broth to make the broth spoilage. Okay. However, with the swan neck, okay, so the pastor also put a piece of cotton in the swan neck. So even the air can get in, however, the bacteria cannot get in. Okay, so this means here is the sterile condition. So even he put this flask for a long time. However, so the broth is still fresh. Okay, so no bacteria grow in the broth. So that means that the microorganism or the bacteria can degrade the organic material. Then let's talk about the microscope. There are two scientists, Robert Hooke, Antony van Leeuwenhoek. So those two scientists invented the microscope. So they use their microscope to observe some materials such as the material scrubbed from their teeth. Okay, so they can see thousands tiny living organism. Okay, then they found the microbe. Okay, so and then they posted the subject of microbiology. Actually, their microscope can make the magnification of 300 times. Okay, it's very close to our microscope. Okay, because our microscope can make 400 to 1000 magnification. Ferdinando Cole discovered the heat resistant Windows pore. Okay, so the Windows pore can make the microorganism very strong and to resist to extreme conditions. Oliver Hormans and Ignaz Semmelweis described the importance of hand washing in the preventing disease in hospital. So those physicians found that the bacteria or the microorganism can cause the diseases in the hospital. So that was the healthcare providers such as doctors and nurses who carry the microorganism to the patients. Okay, and then they suggested that all the nurses and the doctors have to wash their hands before contacting the patients. Okay, so this method can decrease the diseases happened in the patients. Okay, that's why now if we go to the clinic, okay, to see the doctors or the nurses have to wash their hands to do the examination on the patients, right? Joseph Lister used aseptic technology in surgery. So Dr. Lister was a surgeon. Okay, he also found that there were tons of infections after surgery, and then he generated this technology uh, to decrease the chance to transfer the microorganism from the health providers to the patients during the surgery. Okay, actually, this technique has already saved tens of patients. Now, Louis Pasteur has found that the microorganism uh, can contaminate the organic material and degraded the organic material, right? And then, so Louis Pasteur invented technology to sterilize the liquid organic material. So this technique is called pasteurization. Okay, so the pasteurization is widely used to sterilize the liquid products. Okay, such as milk or the juice. Okay, so we still use this technique now. Okay, Robert Koch. So the Dr. Koch postulated a series logic steps that establish whether or not an organism is pathogen and which disease cause. Okay, so Dr. Koch found that the anthrax is caused the Bacillus anthracis in 1875. Now let's talk about the events. So the important events in the biotechnology history. Okay, so the first is the discovery of restriction enzyme. So it is discovered in 1970s. Okay, so the scientist Werner Aber Daniel Nassans discovered the restriction enzyme. So those enzymes actually can cut the DNA sequence. Okay, so we can use this enzyme to cut the DNA sequence. And then we can either remove a fragment of the DNA or insert one fragment of DNA. Okay, so this is very useful for the biotechnology. Okay. And then second is the invention of PCR. PCR means polymerase chain reaction. Okay, so this is also called thermal cycle. Okay, so it is invented in 1980s. So the scientists can use this technology to amplify the DNA fragment to millions copies. Okay, because the DNA or RNA volume is very low. Okay, so it's very difficult for the scientists to study on the low volume DNA or RNA. So they have to amplify the samples to study. Okay, so actually PCR is very important technology. So this technology was granted Nobel Prize. Okay. So the third is small RNA. Small RNA was discovered 2000s. Okay. So the small RNA actually is a very small portion of the RNA families. However, the small RNAs have many important functions. So they can regulate the DNA. Okay, to attenuate the DNA's function. Okay, so it is very important. So the small RNA are very important in bacteria and infected humans. Okay, so they can be a new target for the antimicrobial therapy. And then, so let's talk about the genetic identification of human microbiome. Okay, so actually, so there are tons of microorganisms on the human body. Okay, even on the surface of our skins. Okay, in the colons, in our colons. So there are tons of microorganisms. So even in our mouth, okay, in our nose, so there are tons of microorganisms. So since those microorganisms live with us for a long time, so it's very important to discover the genome map of this microorganism. So the scientists studied this project. So this is the human microbiome project. Okay. So actually, the scientists isolated the microbe from our body. So they culture this microorganism and then do the study. Okay. Actually, the information of this study can provide the new knowledge for the scientist to know and recognize the microorganism in our body, and so this is very important for the health and this study. Now let's take a look at this question. The scientist who built the first simple microscope was. So I give you 5 seconds. So you can go back and take a look at the slides and give me the correct answer. 1, 2, 3, 4, 5. Okay, so the correct answer is C. Okay. Actually, there are two scientists, Hooke and Leeuwenhoek, who invented the microscope. Now, I have covered all the contents of these lectures. So this is the important outcomes. So you guys can feel free to take a look at and review these slides based on these outcomes. Okay. Now let's talk about the micromolecules. Okay. So what are the micromolecules? So macromolecules actually, so they are very large. Okay, so so they have huge molecular weight. Actually, they are very important for the life. Okay, so we have 4 steps. Macromolecules. These are carbohydrates, lipids, proteins, and nucleic acids. Okay. So the monomer actually, so that's a subunit of macromolecules. Polymers, so there's so many monomer link together to form the polymers. So polymers can be the chains of monomers. So here's the diagram shows the macromolecules such as carbohydrates, disaccharide, polysaccharide, okay, like the starch, cellulose, lipids, such as triglycerides. So we talk about this later on. So carbohydrates. So the carbohydrates actually, carbohydrates are made by the carbon and the water, okay. So we have, so we name the carbohydrates with suffix "-os", okay, such as hexose, pentose, glucose, fructose, okay, sucrose. So sucrose is widely used for the table sugar or the cane sugar. Polysaccharides. Carbohydrates actually so this carbohydrates so we can see this carbohydrate that actually they are the monomer, okay, monomer. So they are the monomer. Actually, the monomer linked together to form the polysaccharides, okay. For example, glucose is the 6-carbon sugar. So glucose is one type of monomer. So several or many glucose linked together can form cellulose. Okay, cellulose. So those polysaccharides actually they are used by the living organism as structural material to build the living organism's body. Okay, for example, cellulose can be used. Cellulose can be used by the organism to build the cell wall. Okay, such as the trees. Okay, or many algae. Okay, so the agar. Okay, agar. So agar can be used to build the medium. Okay, so we can use agar to make the culture medium. Okay, chitin actually is found in the fungi. Okay, fungi use the chitin to build their cell wall or the structures. So this diagram shows the structure of polysaccharide. Okay, so we can see actually, so this is the monomer. Okay, so this is one subunit of the sucrose. Okay, so the sucrose link, many sucrose link together to form the polysaccharide. So if two monomer link together, so it is called disaccharides. Now let's talk about the lipids. First, let's talk about the triglyceride. Okay, triglyceride is the major molecules for the storage of the energy. Okay, so as we can see this term, okay, tri means 3. Okay, so that means there are 3 fatty acids. Okay, so it has one glycerol. So we call this structure as the triglyceride. Okay, so it so triglyceride composes one single molecule of glycerol bound to 3 fatty acids. Okay, so the fatty acid can be either saturated or unsaturated. So this is their diagram of the triglyceride. So this is so this is glycerol. So this is glycerol linked to 3 fatty acids. Now let's talk about the phospholipid. Phospholipid is very important because this is material used to build the cellular membrane. Okay, so this is the structure with the double layer. Okay, this so this material have 2 function heads. One is hydrophilic head. Hydrophilic means water loving, so they can embed in water. Okay, so it is negative charge. Another head is hydrophobic. Hydrophobic means water resistance. Actually, it's situated in tail. Okay, so this terminal is uncharged. Okay, That means this terminal can embed in the lipid. Okay. So that means so these molecules can embed its head to the water and embed its tail to the lipid to form the double layer of the membrane, cellular membrane. As we can see, so this is phospholipid. Okay, so this is so this is so this is hydrophilic head. This is hydrophobic tail. Okay. Actually, the hydrophobic tail can link together to form can link together, and the head of the phospholipid can face to outside to embed in the water. Okay, to form the double layer of the phospholipid. So this is the basic structure of cellular membrane. So the third type of lipid are the steroids. Okay, so we have all kinds of steroids. For example, the cholesterol. The cholesterol is one type of very important steroid. Okay, so cholesterol is the material for the cellular to make the hormones. We also have the vaccines. So vaccines can be used by the microorganism to build their cellular wall to be waterproof. Okay, and so the microorganism can use vaccines to resist the extreme conditions. Okay, such as very dry or very hot. Okay, for example, so the tuberculosis or lepers uh, can use vaccines to build their cell wall. So it's difficult to treat those bacteria because they can use this material to resist the medications. Okay, yeah, tuberculosis and leprosy. So this is the diagram of the cellular membrane. Okay, actually, cellular membrane is made of the phospholipid. Also, so there are tons of steroids such as the cholesterol embedded in the cellular membrane. Also, there are many proteins. So those proteins can work as channels of the receptors. So those have very important functions for the cellular functions. Now let's talk about the proteins. Okay, proteins are very important. These are the predominant organic molecules in cells. Okay, so proteins are made of 20 different amino acids. So the amino acids are the subunit of the proteins. Okay. Actually, amino acids are the monomer of a protein. Once amino acids link together, so they can form peptides. Okay, peptides is short fragment of amino acids. So if the several peptides link together, so we have polypeptides. So that's mean polypeptide means so they have at least 20 amino acids length. Okay. So several polypeptides can form together to can link together to form the proteins. Okay. That's why the proteins actually has very complicated structure. Okay, complex structure. So they have four type of structure. The first is the primary structure. Primary structure is actually its order of the amino acids chain. Okay. Secondary structure. Once the certain amino acids certain amino acids linked together by the hydrogen bond, okay, by the hydrogen bond, so they can form, different, so they can form two different structure. One is the alpha helix, another is the beta sheet. Alpha helix. Here we can see, so this is the primary structure, this is peptide, okay. So if two, for example, two amino acids from a different location, so they linked together by the hydrogen bond, okay, by the hydrogen bond. So they can either form the, so they can either form so they can form either alpha helix or the beta sheet, okay, or the beta sheet. So if these two link together, yeah, so we can have the alpha helix. So if these link together, so we have beta sheet, okay. Tertiary structure. Tertiary structure is made by the disulfide bond, okay, disulfide bond. So if the peptide contains cysteine, okay, because cysteine contains a sulfur bond, so they can link together to form the disulfide bond, okay, disulfide bond. So they can make the tertiary structure. So tertiary structure gave the polypeptide 3D structure, okay, 3D structure. So secondary structure gave the polypeptide 2D structure. So since the polypeptide have 3D structure, so the polypeptide may have its biological function. However, it's not enough to perform the complicated biological function. Okay, so several polypeptides can link together to form the quaternary structure. Okay, so the quaternary structure actually is the structure of a protein. Okay, so since several polypeptides with the 3D structure linked together, so they can form a more complicated 3D structure. So that's why the proteins can perform very important biological functions. As we can see this diagram, okay, so this is tertiary structures give the 3D functions. So several polypeptides linked together, so we have the proteins. So since the proteins have the complicated 3D structure, so they have many important functions. Okay, for so based on the functions, so we can divide it. So we can classify the proteins as enzymes. Okay, so the enzyme. So their function is to catalyze the materials or the substance. Okay, so they can perform certain chemical reactions. Okay. The second is antibodies. So those proteins actually they are glycoproteins. So they have the sugar chains. Okay, so they can conjugate it with the antigens or the receptors of the microorganism. Okay, so they can perform their functions as the secondary signaling pathway or attenuation the microorganism's functions. Okay. So the functional 3D form of the proteins is also called as the native state. Okay, so if the proteins have their own 3D structure, they also have the functions. So the structure is called native state. However, this native state can be can be damaged by certain conditions such as heat, acid, or alcohol. Okay, so those conditions can destroy this native state and cause the proteins lose their functions. So this is called denaturation. Okay, denaturation. Denaturation means the proteins lose their native state and their functions. So we use these conditions widely. Okay, for example, we use heat to cook the meat. Okay, once we cook the meat, so the meat changes its morphology. Okay, so so the meat can shrink and give us a good smell. Okay, so we also use alcohol to clean the wound. Okay, to sterilize the skin, right? Now let's talk about the nucleic acid. Okay, nucleic acid. Nucleic acid. Nucleic acid's function is to be involved in the cellular information storage or transfer. Okay, so nucleic acid contains DNA and RNA. DNA is organic materials that code the genetic informations. Okay, so it's very important for the heredity. RNA actually is used to transfer the genetic information from DNA to produce proteins. Okay, to produce proteins. So translate the DNA information to produce proteins. Okay. Now let's talk about the DNA. Okay. So DNA is formed by two very long nucleotide strings. Okay. So these two strings linked together by the hydrogen bonds. So the this nucleotide string contains four nitrogen base. These are adenine and thymine, guanine, and cytosine. Okay, adenine always paired with thymine. Guanine always pairs with cytosine. As we can see this diagram. Okay, so this is double string, double nucleated string. Okay, so these are the so these are the so these are the nitrogen base. Okay, nitrogen base. So the nitrogen base can pair together. Okay, so this is DNA structure. As we can see, so the DNA is double string. Okay, however, the RNA is single string. RNA is single string. RNA is a single string molecule. So RNA is synthesized as the copy of one strand of DNA. Okay, that means they carry the information of one DNA strand. Okay, and so this RNA is called as messenger RNA because this RNA carries the information of DNA. So the information will be used to synthesize the protein. Okay. So we also have tRNA. tRNA means transfer RNA. Okay. So they can deliver the amino acids to synthesize the protein. That's why they call the transfer RNA. RNA is the subunits of ribosome. So they can link either with mRNA or tRNA. So they can translate the information in the mRNA and use this information to build the amino acids transferred from tRNA. Okay, so those RNAs work together to make proteins. Now let's talk about another macromolecules. It is adenosine triphosphate. It is also called as ATP. Adenosine triphosphate is made with the adenine and ribose. Okay, also three high energy phosphate molecules. Okay. So this is the molecule to transfer energy or supply energy for our body to use. Okay. So once energy is released from the chemical reactions, such as degradation of the degradation of the sugars, so this energy will be released and transferred to the ATP. Then ATP can be used by the cells. Okay, can be used by the cells. So cells can have the energy to perform its functions. So as we can see this diagram. So this is ATP. Okay, ATP can be degraded to ADP, and phosphate. So the energy of the phosphate can be released for cell to use. Now let's talk about the characteristics of cells. Okay. So the cells. So we have three types of cells: bacteria, precardial cells, or eukaryotic cells. Okay. So those cells have their characteristics, such as they have their own specific shape. Their shape could be spherical, polygonal, or the cylindric. Okay. So they also have their specific cellular membrane. Okay. So they have their DNA contains specific genetic information for the cellular functions. Now let's talk about the eukaryotic cells. So eukaryotic cells could be the animal cells, plant cells, fungi cells, or the protozoa. Okay. So eukaryotic cells have the true real nucleus. Okay. They also have organelles. Okay. So these organelles can be used to produce the proteins of the hormones. Okay. bacteria and archaea, so they are precursor cells. So they don't have nucleus. So they are relatively smaller than the eukaryotic cells. So they don't have organelles. Okay. So their structure are simple. Okay. Now let's work on this question. Cell membranes are composed of what? So I gave you guys 5 seconds. You guys can review the previous slides and give me the correct answer. 1, 2, 3, 4, 5. So the correct answer is C. Phospholipids. Okay. So here is the. Now let's talk about the next section. Okay. So we are going to learn the knowledge how to classify the microorganism. Okay. So the science subject to classify the living organism is called taxonomy. Okay, so it's a science for the classifying, because since the scientists have discovered so many things. Okay, so many bacteria, so many eukaryotic cells. So the scientists love to classify and classify those things. Okay, so they generated this subject, taxonomy, developed by the Carbon Lean. Okay, 1700. Nomenclature is the assignment of a scientific name to various taxonomic categories to the individual organism. Okay, classification means the orderly assignment of organism into hierarchy. Okay, identification. So if the scientists discover many living organisms, so they have to do some study to identify those living organisms. So since they found if they found the similar characteristics of those living organisms, so they can classify those living organisms to one category, right? So this is identification. Now let's talk about the nomenclature. Okay. So this is assignments of so this is assignment of scientific names of the living organism. Okay, so that's so the scientists try to give the name to the living organism. Okay, so actually the scientists gave two names for one living organism. So this is called binomial system. Okay, binomial system. That means the scientists gave two names to one living organism. It's like our human. Our human have two names. Okay, at least two names. Okay, first name and last name, right? So the binomial system is a combination of the genus and the species name. Okay. So the scientific names are the italicized. Okay, italicized. When they are written in print and underlined, when they are written by the hand. Okay. It's like E. coli. Okay, E. coli. Okay. So the E. So the E. coli actually So the Escherichia is the genus name. Okay, coli is the species name. Okay, so we can abbreviate the last name. Okay, abbreviate the genus name. So we can abbreviate this name to E. coli. So we have for the classification, we can classify the living organism to these 8 categories. Okay, domain, kingdom, class, order. Okay, species. So the range of this category is from big to small. Okay, from domain to species is smaller and smaller. Actually, for our microbiologic studies, so we are interested in the genus and the species. Okay, so we can focus on that. So this is domain. So we can categorize to the eukaryotic and prekaryotic cell. Okay. Now let's talk about the evolution of microorganisms. So the phylogeny, okay, phylogeny is the taxonomic scheme that represents the natural relatedness between the group of living beings. Okay, so the evolution means the hereditary information of living beings change through times. Okay, so this theory is generated by by Darwin. Okay, Darwin. So Darwin's theory is natural selection. Okay, that means the natural can change the can change the function of living organism, and these functions can be transferred to their next generations. So the Darwin and Haeckel proposed two kingdoms, the plants and animals. Haeckel's later added another kingdom. This is protozoa. Okay. Now let's talk about the Wolff-Fox system of taxonomy. Okay. So this system is based on the conserved small subunit ribosome RNA sequence. Okay, because before the scientists classify and categorize the living organism based on the phenotype of the living organism, such as their hair color, skin color, or the shape of the organs. Okay, and so since the scientists have the newer technology such as the PCR, so they can do the sequencing on DNA or RNA. Okay, so if they sequencing the ribosome RNA, so they can use the information of the ribosome RNA to categorize the living organism. So with this technology, so they can the scientists can categorize 3 domains: bacteria, archaea, and eukarya. Now let's look at this question. Which of the following are the domains of the Wolff-Fox system of taxonomy? So I give you 5 seconds. So you can review the slides, give me the correct answers. 1, 2, 3, 4, 5. Yes, we can see the. So the correct answer should be B, C, D. Okay. BCT. It's in this slides. So this is appendix, okay? Okay, so I have covered all the contents of these chapters. So you guys can feel free to review the contents of these chapters based on this appendix, okay? So this is end. Thank you for your attention.